This lesson is about biological treatment method used in wastewater treatment. Now, all of you are familiar with the conventional wastewater treatment plant. This is a sketch of conventional wastewater treatment plant. As you know, wastewater treatment plants begin with the preliminary treatment where screeners, grid chambers, and skimming tanks are available. After the preliminary treatments, there are primary treatment method which consist of coagulation tanks, flocculation tank, and primary clarifier. Then we can find the secondary treatment units. After the secondary treatment unit, you can find tertiary treatment units or advanced treatment methods. Tertiary treatment method consists of disinfection, nutrient removals. Advanced wastewater removal method includes salt removal such as reverse osmosis, electrodialysis, ultrafiltration or microfiltration, and activated carbon adsorption. In secondary treatment, you can find aeration tank and secondary clarifier. So both these components are called as activated sludge wastewater treatment. So this is called as activated sludge process because in activated sludge process, the sludge coming from the secondary clarifier, the part of the sludge coming from the secondary clarifier is recycled back to the aeration tank. So here there are microorganisms in the secondary sludge. Because of that, we can call it as activated sludge. It is activated used from microorganisms. So for the aeration tank, for the aerobic digestion reaction, microorganisms are required. Microorganisms do the reaction so that the microorganisms available in base sludge is recycled back to the aeration tank. Part of the sludge is recycled. Let's see what is biological wastewater treatment. Now already you know what is the meaning of biological wastewater treatment. So if you understood the activated sludge process, then you can easily understand the other biological wastewater treatment processes. Okay. We can treat wastewater containing biodegradable constituents using the biological wastewater treatment. So only biodegradable constituents can be treated using biological wastewater. So in activated sludge process, we can remove dissolved organic matter and also the colloidal suspended matter, and both should be inorganic matter. So if they are inorganic, like heavy metals, we cannot use activated sludge process or any other biological treatment method to remove the inorganic waste from the water. So what are the objectives of biological wastewater? The first thing is to remove the biodegradable component from the wastewater. So we can say the first objective is oxidize, dissolve and particulate biodegradable constituents. The particulate mean that colloidal and suspended solids. So oxidize, dissolve and particulate constituents into acceptable end product. So as you know that according to the equation, the organic matters are converted into simple end products. That means CO2, H2O and other non-degradable part. Then again we can say another objective is to capture and incorporate suspended non-settleable colloidal solids into biological block or biofilm. Okay. So there are settleable solids. There are biologically there are settleable solids. Then, so there may be non-settleable solids. So non-settleable solids also capture and incorporated into biological block or biofilm. Then next one is uh, transform or remove nutrients such as nitrogen and phosphorus, because it is required to remove nitrogen and phosphorus of the wastewater. If the wastewater is discharged into the environment, to especially to a water body, if there are nitrogen and phosphorus, those water bodies can get eutrophicated. Eutrophication can be formed. To avoid that, nitrogen and phosphorus, those nutrient has to be removed. In some cases, remove specific trace organic constituents and compounds. So, now we know from biological wastewater treatment method, we can remove dissolve and particulate carbonaceous BOD. So now you all of you know BOD is the amount of oxygen required to degrade the organic matter biologically. So that here this represent removal of dissolved and particulate carbonaceous organic matter. And stabilization of organic matter, here OM means organic matter, found in wastewater is accomplished biologically using variety of inner microorganisms, principally bacteria. So organic matters are converted into simple products and non-degradable parts. So that it is the meaning of stabilization of organic matter. Then microorganisms are used to oxidize the dissolved and particulate carbonaceous organic matter into simple end product and additional biomass. There may be non-degradable additional biomass. So the purpose of biological wastewater treatment is to aerobic biological oxidation of organic matter. So this is the reaction involved with the aerobic biological oxidation of organic matter. So here you can see organic materials are converted by microorganisms in the environment oxygen, and there may be ammonia and phosphorus also like nutrients. So the microorganism converted this organic material into CO2, H2O and new cells. So new cells represents the microorganism population also, because the microorganisms eat the organic material, then their population grow. Their population is increased. The term new cells is used to represent the biomass produced as a result of oxidation of the organic matter. So this, the term new cells is the biomass. So this may be consist of microorganism plus those degradable part of the organic matter. Microorganisms are also used to remove nitrogen, phosphorus in wastewater treatment processes using anoxic process, using nitrification or denitrification process. Specific bacteria capable of oxidizing ammonia to nitrite, nitrate, while other bacteria can reduce the oxidized nitrogen to gaseous nitrogen. Okay, so for this I will share you a handout. You can refer them about the nitrification process and the nitrification process in wastewater treatment. So you may remember, so this is part of a conventional wastewater treatment plant. So here bars track, grid chamber, fat trap, then primary clarifier. So after the primary treatment, the water is sent to aeration tank. Then after that, in aeration tank, microorganisms do the job. They digest the organic matter. Then it is sent to secondary clarifier. Then secondary sludge is recycled. Part of secondary sludge is recycled back to the aeration tank. So this the both two components are called as activated sludge process. Okay. And this is the whole diagram of the conventional wastewater treatment plant. I am not going to talk about this. Preliminary treatment, primary treatment, secondary treatment, advanced treatment, and tertiary treatment. So usually in secondary treatment and tertiary treatment, we can use biological treatment methods. So there are two types of biological wastewater treatment processes: aerobic biological oxidation and anaerobic biological oxidation. So here you can see anaerobic mean anaerobic. Anaerobic, that means there is no air. Aerobic mean with air, there are oxygen. So aerobic biological oxidation process can be divided into suspended growth process and attached growth process. So let's see what are aerobic biological oxidation. So the simple example for aerobic biological oxidation is the activated sludge process. It is a suspended solid. It is a suspended growth process. So aerobic biological oxidation remove organic constituents and compound to prevent excessive dissolved oxygen depletion in receiving water. What does that mean? So if there are organic matter in the wastewater, what happen? The dissolved oxygen level is decreasing because the microorganisms consume the dissolved oxygen level to degrade that organic matter. So by aerobic biological oxidation of wastewater, we can remove the organic matter. So once they discharge into a inland water body or whatever place, then no need to worry about the dissolved oxygen depletion because there are no microorganisms in the wastewater. There are no organic matter. All the microorganisms has consumed the organic matter. Then it will reduce the dissolved oxygen depletion. Then, so here the main purpose is to remove organic constituents. Then remove colloidal and suspended solids to avoid accumulation of sludge. So these are the objectives of aerobic biological oxidation. Okay. So my dear students, no need to cramp all these things. No need to remind all these things. You just understand what is the purpose of aerobic process and anaerobic process. Okay. Then remove colloidal and suspended solids. Reduce concentration of pathogenic organisms. Remove BOD. So that means BOD is the organic matter, amount of oxygen required to degrade the organic matter biologically. So we can reduce the BOD value by aerobic biological oxidation. Then SGP, the suspended growth process, and attached growth process requires sufficient contact time between wastewater and microorganism and sufficient oxygen and nutrients. So here SGP, suspended growth process, and attached growth process require sufficient contact time between wastewater and microorganisms, and there should be oxygen also because these are aerobic. There are some problems encountered in aerobic processes such as bulking sludge when biological sludge has poor settling condition. So the primary sludge is it can be easy to settle, but bulking sludge it is not in biological sludge it is not easy to settle. For a primary sedimentation tank we can put coagulant and flocculant, But for the biological treatment processes, we cannot put coagulant and flocculant to the sedimentation tank. So sometimes it will inhibit the microorganisms. So the coagulant and flocculant can be toxic to the microorganisms. So there is a poor settling condition in biosolids. And forming, the forming means the forms, spinner, are formed. Development of two bacteria generally which have hydrophobic cell surfaces and attached to air bubble surfaces where they stabilize the bubbles to cause form. So forming is another problem. Because of that, the air will not come from the atmosphere to the water. So it is a problem. Then here this is the reaction available in uh, aerobic oxidation of organic matter in a wastewater. So in previous you learned that the one equation. So that equation can be categorized into two steps. So the first step is oxidation and synthesis. So here organic matter plus oxygen and nutrients, then microorganisms convert the organic matter into CO2, ammonia, other end products, and C5H7NO2. So these are the new cells that we call. Then what happened? These new cells react with oxygen by microorganism and create CO2, H2O, ammonia, and end products. So here nutrients are ammonia or phosphorus. So here COHN is the organic matter. So you have to remember these two equations, and you can understand that organic matter, so this is the organic matter plus oxygen plus nutrients and bacteria are converted to CO2, H2O, ammonia, and end product. So this is the aerobic biological reaction. So these are intermediate product, new cells. So for aerobic biological processes, the pH should be between 6 to 9. Optimum performance occur near neutral pH. A reactor dissolved oxygen concentration should be 2 milligram per liter is commonly used. That means aeration tank should have DO level 2 milligram per liter. Then you heard that there are two types, suspended growth process and attached growth process. Activated sludge process is an example for suspended growth process. So in activated sludge process, in aeration tank, what happened? The microorganisms are suspended. Microorganisms are available as a suspension. Okay, suspension. They are suspended in the water. Vathura ellila innawa. So microorganisms responsible for the conversion of organic matter in the wastewater to gases and cells tissues are maintained in suspension within the liquid by appropriate mixing method. So in aeration tank there are appropriate mixing method. So it's the mixing method may be given by supplying compressed air or using mechanical impellers. So this is again a sketch of activated sludge process or suspended growth process. Mechanical equipment is used to provide mixing and transfer of oxygen into the process. In aeration tank, contact time is provided for mixing and aerating influent wastewater with the microbial suspension. The mixed liquor then flows to a clarifier where microbial suspension is settled and thickened. And remember this term mixed liquor. So mixed liquor of wastewater means the liquid or the water in the aeration tank, mixed liquor. So after mixing and aerating influent wastewater with the microbial suspension in the aeration tank, so this mixed liquor is sent to the settling tank. Settled biomass described as activated sludge. Settled biomass, the biomass settled at the bottom of the secondary clarifier is called as activated sludge because of the presence of active microorganisms. Right? Then these activated microorganisms is recycled back to the aeration tank. The portion of thickened solid is removed daily or periodically as the process produces excess biomass that would accumulate along with the non-biodegradable solid contained in the influent wastewater. Okay, so the part of the sludge is recycled, that another part is disposed with the other solid. So this is a picture of aeration tank, and here you can see the air distribution system here. Then this is another compressed air lines in aeration tank, and these are aeration basin of one of wastewater treatment plant. So here some impellers are used. So impeller, the mechanical aeration system can be divided as fixed surface aeration or floating surface aeration. Here, those impellers, mixing impellers, turbine, agitators are fixed to one point. And some places there are floating surface aerator. They are floating. They are not just fixed to a fixed point. So these are floating. Here, this is a fixed one. Okay, you can see this diagram and try to understand what happened in this activated wastewater treatment plant. Let's see what happened. So this is the influent for the wastewater treatment plant. The wastewater is coming from a domestic sector, or we can assume they are coming from industries also. Then first unit operation is clean, the second one grit removal, then there's a primary clarifier, aeration tank, then secondary clarifier. Then you can see the return air activated sludge. Activated sludge is recycled to the aeration tank, and a part of the sludge is sent to the sludge settle system. Here the primary sludge is also coming here to the sludge storage tank. Then that sludge is sent through belt press, filter press, you already know. Then they are sent for bioset. So that bioset may be composting or anaerobic digestion, or maybe biogas degradation. Then after that, the residue is sent for the landfilling, and part of them is sent for incineration. So usually if there are very high toxic things like heavy metals, then they can be sent to incineration. So in my previous lecture also, I told you that in Sri Lanka also, in our country, some of wastewater treatment plant sludge are disposed in a cement kiln, in Siam cement factory in Puttalam. As example, in textile factories, wastewater sludge may contain some heavy metals. So during the dyeing process, coloring process, some heavy metals are added into the wastewater. Then they are settle at the with the sludge. So that sludge is the moisture of the sludge is removed from sun drying or any other method. After that, that sludge is sent to incinerate in the cement kiln of Siam City factory. So this is already available in many other countries. So not only the waste water treatment plant sludge, but also many type of waste are disposed in cement kiln, such as outdated medicine, some pharmaceutical drugs. You may have heard during the Maithripala Sirisena president's era that drugs were disposed in the cement kiln. Then some textile waste, some the consumable waste are disposed in cement kiln. Then let's see what is attached growth process. Microorganisms in attached growth process, microorganisms are attached to some inert medium. That means there is a packing material. now you can understand in in suspended growth process, microorganisms are suspended in the water. වතුරේ එල්ලිලා ඉන්නවා. But in attached growth process, microorganisms are මොකක් හරි inert material එකක ඇලිලා ඉන්නේ. So those inert material are racks, slags, stones, rocks, specially designed ceramic or plastic materials. The packing can be submerged completely in liquid or not submerged. Okay, so that මේක වතුර කෙලින්ම ගිලිලා තියෙන්නත් පුළුවන්, බාගෙට ගිලිලා තියෙන්නත් පුළුවන්. Attached growth processes are known as fixed film processes. Okay, this is a similar term. Examples are trickling filters and rotating biological contactors. Let's see what is a trickling filter. So before I read this, you can see this is a trickling filter. So influent, the wastewater comes from this inlet. So I told you that these biological methods are applied in secondary treatment or in tertiary treatment. So after the primary treatment, after the primary sedimentation tank, the clarified water is sent to the trickling filter. This is the inlet for trickling filter. So influent is coming through here and it goes up. So at the end of at the top, the water is distributed uniformly. So there are special hoses or nozzles here. So here there's a shaft here. So this shaft can be rotated. Because of that, water can be evenly distributed while it is rotating. So anyway, what happened to the microorganism? Where are microorganisms? So here, this is the media. That means inert materials are available here. So those racks, slag stones are placed here. Then microorganisms are attached to that packing material. You may remember in distillation also you learned that there are some internals, trays and plates and also some packing materials. So likewise, there are packing. It may be stone. Okay? So then microorganisms are attached to the surface of that stone. Then what happens? The wastewater is flowing. The wastewater is flowing from top to the bottom. So while wastewater is flowing from top to the bottom, the wastewater contact with the microorganism who were attached to the inert material. Then what happened? During that time, the microorganisms eat the organic matter in the wastewater and converted it to simple end products. So at the bottom, we can get organic matter free water. But that water may contain some biosludge. Because of that, because of the presence of biosolid, we have to send this water to a sedimentation tank. So this is the secondary clarifier in a conventional wastewater treatment plant. Then we can remove the sludge and we can send it to the sludge treatment. Then we can get clarified water from the top of the tank. So this clarified water then we can send into the tertiary treatment units after disinfecting. Then usually the part of the treated water is recycled back to the filtration tank for further purification. And you know this is a aerobic biological reaction. That means there are organic matter, CHONS. Then microorganisms do the job with the presence of oxygen. So where are oxygen? So it is better to give additional oxygen. Here, this is the airflow. So by using a blower or compressor, we can provide extra oxygen. So these are some pictures of trickling filter. Here, uh, this is the top of the trickling filter. So this is available underneath, beneath the floor. So this is the way that water distribute uniformly through the whole surface. So while it is rotating, while this shaft rotating, this water uniformly distribute throughout the area of the trickling filter. Then this is, let's see here, this is the effluent inlet. So the wastewater coming after the primary sedimentation tank enter into the trickling filter, and here it enters from here. Then at the top of the trickling filter, it uniformly distributes throughout the cross-sectional area of the wastewater uh, filter. Then there's a blower to enter air, blower. So this is the blower entrance. So this is the blower entrance. Blower entrance. So the air will go into the trickling filter. So what happened here? When wastewater going down, it will contact with the media, and microorganisms are attached to the media. Then microorganisms and wastewater contact with each other, and they do the aerobic biological oxidation. Then from here, the treated water is going out. So if we want, we can recycle the treated water again to the again with the influent. So let's see what is about trickling filter. So trickling filter is a secondary treatment process, and therefore follows primary treatment. So the trickling filter usually located after the primary treatment or primary sedimentation tank. Then primary treatment removes settleable and potable solids. So by the time wastewater reaches the trickling filter, it contains mostly colloidal and dissolved solids. The purpose of the trickling filter is to remove these colloidal and dissolved solids through the aerobic biological decomposition process. In the trickling filter process, the wastewater is spread down over the microorganisms and is allowed to trickle down to the microorganism covered media. Trickle down or allowed to flow down. In trickling filter, wastewater is distributed to the top area of the vessel containing non-submerged packing material. So this can be either submerged or non-submerged. Historically, rock was used most commonly as the packing material. So that we can say that දියසෙවල සහිත ගල් rock. The most modern trickling filters vary in height from 5 to 10 meters, filled with plastic packing material for biofilm attachment. So there may be plastic film material also. So we have to add nutrients, example nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, iron, zinc, manganese, nickel, like that. We have to add as the nutrients for microorganisms. So now you are familiar with this diagram. So here, but there's a difference here. So we can replace activated sludge process with trickling filter. That means we can replace aeration tank with the trickling filter. But here you can see there is no recycle of activated sludge. The secondary sludge is only taken away. Then next type of attached growth process is rotating biological contactor, RBC. In the rotating biological contactor RBC process, the media is in the form of drum. Microorganisms grow on this drum. Drum is slowly rotated periodically, submerging the microorganism in the wastewater, where they are able to remove organic materials from the wastewater for food. Drum is slowly rotated, periodically submerging the microorganisms in the wastewater. Let's see. So this is the rotating biological contactor here. So the microorganisms grow on the surface of the drum. Okay. Then what happened? The drum is slowly rotated, periodically submerging the microorganisms. So here look at this is the wastewater holding tank. Wastewater holding tank is this one. So it is not full of water. It is filled with half of water. Okay, it is filled. That means there are water. That means this part is wetted with water. Then that's why it is called as drum is slowly rotated, periodically submerging the microorganisms. So part of the drum is submerged. Drum එකෙන් භාගයක් තමයි ගිලිලා තියෙන්නේ. The part of the drum is submerged in the wastewater. Therefore, microorganisms, their microorganisms are attached to the surface of the drum, but the part of the drum is submerged. Therefore, half of the population of microorganisms are contacted with the wastewater. Now they are under the water. So when it is rotated, the microorganisms who are under the water comes to the top. That means there is no wastewater. Then what happened? The microorganisms will take oxygen from the atmosphere. Then they do the aerobic biological oxidation reaction. That means now the microorganisms that the surface of the drum is wetted with wastewater, and surface is attached with microorganisms. So now oxygen is coming from atmosphere. So now all three ingredients are there: organic matter, microorganisms, and oxygen. Then they convert the organic matter into simple end products, CO2, H2O, and other products. Then there should be a stopping mechanism or cutting mechanism to remove the microorganisms with new cells. So likewise, we can rotate this RBC periodically. So usually rotating biological contactors can be arranged like series, like this in way. Then after the RBC process, there should be a secondary settling tank. Then this sludge is not recycled as in activated sludge process. Okay, so you must remember this sludge is sent to the sludge disposal unit. So these are some pictures of rotating biological contactors. Actually, it is not a full cylinder. This cylinder is made of, the cylindrical configuration is made of using several discs. Okay. So before you see the some pictures of RBC, now you can understand. So we can replace the aeration tank with RBC, and there is no return secondary sludge or recycling of activated sludge. So we can replace activated sludge process using RBC, rotating biological contactors, or using trickling filter. That means we can replace aeration tank with RBC or trickling filter. And the important thing is there is no recycling of activated sludge or secondary sludge as in activated sludge process. Okay, so let's see pictures of rotating biological contactors. So there are some advantages of RPC process. It is simple, low energy requirement, nitrification, few nuisance, wide flow range, and large biological population. Handle shock loads, low head loss. Then there are disadvantages: limited control, enclosures, limited experience and training. So these are the components of a rotating biological contactor. There are contactors, disc, tank, and there should be a clarifier. So these are the contactor is made from shaft and disc. Yeah, contactor is made from shaft and disc. So that disc is made of media such as high density polyethylene. So or we can use carbon black also. So so there are corrugated irregular surface in the contact disc. So since it has irregular corrugated surface, the microorganisms can attach to the surface, disc surface. Then this surface are called as dimple surface. And this one is, you can find, this is dimpled. Okay. Then this is some pictures of rotating biological contactors. Here it is maintain some gaps here for operational or maintenance purposes. So then this is the contact disc. So this is the disc. Then biomass are grown on the disc. Biomass, it's look like DSL. So between two discs, there should be at least 1 inch of space. Then usually RBCs has 10 to 12 feet diameter. Some of them are 20 feet long. Okay, 25 feet long. Then media, this is standard density media, 1 inch, high density media. Then this is high density. The shaft has several configuration. So this is a square shaft. The shape of the shaft is square. And this is a round shaft. And octagonal shafts are also available. Octagonal shafts, here you can see octagonal shaft. Then bearings also need. So some mechanical part is also available. And there are drive system, chain, motors. So direct drive systems. So my dear students, I show you this picture for your extra knowledge. No need to cram these things for your exam, but you should know what is RBC, what is trickling filter. Let's see anaerobic digestion method or anaerobic biological oxidation. Okay, biological processes in wastewater can be divided into two: aerobic oxidation and anaerobic oxidation. So simply we can say anaerobic oxidation is without air. Without air, microorganisms convert biomass into a simple end product. So the anaerobic oxidation example is the biogas production. Okay. So anaerobic oxidation is used primarily for the treatment of waste sludge and high strength organic waste. So usually anaerobic oxidation is used for the treatment of sludge. Okay. and high strength organic waste. There should be high strength organic waste. The organic content should be very high and the BOD value should be very high. And there are advantages. It lower biomass yield. From anaerobic oxidation, we can the residue of the sludge is very small compared to the aerobic oxidation. So from aerobic oxidation process, after aerobic oxidation process, that means after aeration tank, trickling filter, RBC, there should be a secondary clarifier to treat the sludge. The sludge has to be treated, separated from the clarified water. But in anaerobic oxidation, the lower biomass yield, there's a lower biomass yield, is the production of sludge is very low compared to the aerobic method. Then we can recover energy from aerobic oxidation method. We can recover energy in the form of methane. Then it can be operated in mesophilic temperature because mesophilic bacteria do this job. For treating high strength industrial wastewater, anaerobic treatment has been shown to provide very cost effective alternative to aerobic processes with saving in energy, nutrient addition and reactor volume. So when we talk about aerobic process, trickling filter, RBC or activated sludge, they need energy to run impellers, to give compress air, like that energy is required. But for anaerobic oxidation, the amount of energy requirement is very low, and nutrient addition is also low because for aerobic tank, nutrient has to be provided for the survival of microorganism, and also reactor volume is very high. Anaerobic oxidation is a complex biochemical reaction carried out in a number of steps by several ways. Several types of microorganisms that require little or no oxygen to live. During this process, gas that is mainly composed of methane and carbon dioxide, also referred to biogas, is produced. From biological anaerobic digestion, methane and carbon dioxide is produced. This is called as the biogas. The amount of gas produced varies with the amount of organic waste fed to the digester, and temperature influences the rate of decomposition and gas production. So in anaerobic digestion process, there are 4 steps. First, hydrolysis occurs, then fermentation occurred, acidogenesis occurred, then finally methogenesis occurred. That means in hydrolysis, complex organic matter is decomposed into simple soluble organic molecules using water to split the chemical bonds between the substances. That means that polysaccharides, carbohydrates are converted into simple matters like glucose. Then fermentation, the chemical decomposition of carbohydrates by enzyme, bacteria, yeast or molds in the absence of oxygen, the passive. Then acidogenesis, the fermentation products are converted into acetate, hydrogen and carbon dioxide by what are known as acidogenic bacteria. So this is done by acidogenic bacteria. This is done by acidogenic bacteria. This is aceto. Then last step is methanogenesis, is done by methanogenic bacteria. So for anaerobic digestion, there is a wide set of bacteria. So these are the anaerobic digestion steps in simple way. The first, the carbohydrate, protein, fats are converted into sugar, amino acid, fatty acids respectively. Then they are converted to volatile acid by fermentation process. Then from acidogenesis, these volatile acids are converted to acetic acid, H2 and CO2. Then methanogenesis, they are converted to methane and CO2. So again here you can see the patterns of carbon flow in anaerobic digestion. So how the complex organic matters are converted to methane and CO2, and there are other non-residue part also. Then here this is a summary. Aerobic digestion, organic matter uh, are converted by microorganism with the oxygen environment to CO2, H2O plus biomass and new cells. Then these biomass and new cells are converted to CO2, H2O and cell debris by the microorganism with the oxygen. So if it is a nitrification process, I hope you are familiar with the nitrogen cycle. Nitrification process, ammonia plus iron is converted to nitrate and biomass new cells with the presence of oxygen by microorganism. The phosphorus immobilization means if there are phosphorus in the cells, so they react with microorganism, the microorganism with the oxygen environment, those phosphorus are converted to biomass or new polyphosphate. Then there's another process called denitrification. It is an anoxic digestion. Anoxic mean denitrification. Here the nitrate are converted to nitrogen by the microorganisms. Then anaerobic digestion. So here acetate and polyphosphates are converted to PHP by microorganisms. Then again my organic matter are converted to methane, CO2, H2O and biomass new cells. So this is the uh, reaction that you have to remember. This is the complete reaction relevant to the anaerobic digestion. So I add these just for your information. Here you can see this is on the carbon elimination. So not only carbon elimination, for nitrification, that means ammonia are converted into nitrate by aerobic tank. Then what happened here? Denitrification, that means nitrate are converted to uh, nitrogen. So it is here denitrification, nitrate are converted to nitrogen. So this is denitrification. Then biophosphorus removal. So when we talk about bacterial growth and substrate utilization curve, so don't worry, no need to worry about this bacterial growth. Okay, so this is the lag phase. Bacteria get adjusted to new environment. Lag phase. And then log growth phase. So this is the lag phase. Then this is the log growth phase. Exponential growth phase. Stationary phase. So this is the stationary phase. The microorganism's growth rate is stationary. Death phase. Bacteria die faster than the reproduction. So this is the bacterial growth curve, and substrate utilization curve is this one. What is substrate? Substrate utilization curve is this one. What is substrate here? The substrate is the food for the microorganisms. We can say BOD, or we can say the organic matter concentration, or the COD. Actually it should be BOD because this is microorganisms. So this at lag phase, the BOD value is slightly increases. So during the exponential growth phase, the BOD value reduces drastically. And in stationary phase and in death phase, the BOD value does not change. Okay, so this is the end of the biological wastewater treatment, and also this is the last lecture of the wastewater treatment.